So today I'm going to be talking about brainwaves and movement with particular emphasis on the idea of phase. And if you don't know what phase is yet, don't worry, I'm going to talk about that. I just recently started my own lab at the University of Texas. And like we said, I was previously at the NINDS. So most of the work I'm going to talk about, I actually completed during my postdoc. There's a little bit at the very end that I've done in my own lab. And then the other thing I want to say is a lot of what I'm going to talk about today is related to basic mechanisms of movement and motor control, but all with sort of an eye towards stroke rehabilitation and helping people who have had strokes regain movement control. That's sort of the long-term goal of this work. So without further ado, let's dive in. So the human brain is capable of controlling voluntary movement quite nicely across a variety of joints, a multitude of muscles. And this allows us to do basic activities like typing on a keyboard, which of course in COVID times, we're all doing a lot more than usual. Also inserting keys into locks so that we can enter our homes, as well as even buttoning up our shirt sleeves. And many of these actions are pretty easy for healthy people without movement impairments to do, perhaps you or I. But for people who have had a stroke, it's not so easy. And stroke is a leading cause of long-term disability throughout the world. And after stroke, what often occurs is that some people will spontaneously have some motor recovery. So they begin to regain their ability to control movements, but that recovery is usually not complete. And this leads to about 40% of people, even at the chronic stage, so 6 months out, still having hemiparesis, which means weakness or paralysis on one side of the body, contralateral to the side of the brain that had the stroke. And over the last few years or the last decades, there's been a surge of interest in this technique called transcranial magnetic stimulation. And the idea with this technique, or one of the original things we wanted to do with it as a field, was use it to help people who have had strokes recover their movements better. So I'm just going to review a little bit of the basics of TMS to make sure we're all on the same page. Basically, TMS, we just hold a wire coil over the head, and we usually hold it flush with the scalp. And inside this coil, we have all these wires that are wrapped up. And when we send a current through those wires, that generates a magnetic field that can pass through the brain or through the scalp and the skull without damaging it, so that it activates some of the cells in the brain. And when we hold this TMS coil over the motor cortex, we see in the periphery, so in the muscles, perhaps in the hand or the arms, we see these little muscle twitches, and that's what is shown here. So this is basically a recording using EMG, which records muscle activity from the surface of the skin overlying muscle. And we can see that after we give a TMS pulse, so a pulse of this, we see this little blip in that EMG signal. And this is what we call a motor evoked potential. And when we activate the motor cortex using TMS, we activate the corticospinal tract. And this tract originates from sensory motor cortical areas, descends down all the way through the brainstem, crosses over, and then synapses on alpha motor neurons in the spinal cord. Those motor neurons are the ones that project out to our muscles. And so when we activate this tract using TMS, we end up activating the whole thing, and then it leads to this twitch at the muscle level. And the reason I'm going into some of the details of this is that this is sort of our baseline measurement of saying, how functional is this tract? Is this tract intact? Is it working properly? The reason that's of interest for us for movement is that this is the tract that is the primary contributor to voluntary skilled movement, especially in the hands in humans. And it's also often disrupted after stroke. So people who have had a stroke at anywhere along this pathway can end up damaging that pathway, and then that leads to poor ability to control movement. So what we can do is we can look at the size of this MEP, or if it's there at all, and say whether or not that tract is intact in a healthy person or a patient who has had a stroke. And so all of this helps us probe the integrity of this corticospinal tract that's so crucial for movement, but it doesn't really help us change the function of the corticospinal tract. So what we can do to do that is we can deliver this TMS in a specific pattern in order to increase the function of this corticospinal tract, or to, in some cases, decrease the function. And this has led to a whole field of TMS research that tries to use transcranial magnetic stimulation to upregulate corticospinal tract function or downregulate corticospinal tract function. So here I want to get to sort of the crux of the issue that I'm going to try to tackle going forward. So this is a figure from a study by Virginia Lopez Alonso, and she did two different TMS interventions. And it doesn't really matter what the details of these TMS interventions are, just know that the goal is to increase the size of that MEP that I showed you so that we're increasing the function of the corticospinal tract. And what she showed is that actually, when you do this in healthy people who haven't even had a stroke, so they have much more intact brains, of course, you can end up seeing that only or less than half of the participants that you will apply the stimulation to, you actually see the expected response. So people tend to fall into what we call responders in black here and non-responders in gray. And the thing that really jumps out to me with this figure is that more than half of the participants, the healthy participants that they tested, were actually non-responders. And so if we want to use this type of TMS to upregulate corticospinal tract function, it's sort of problematic, right? If we can't even get it to work in all, or at least the majority of the healthy people we're testing, then that is probably not going to work so well once we try to translate that to a stroke patient. And this has led this whole sort of problem encapsulated in this figure is often called the variability problem. So people in the field of TMS refer to this when they're talking about not being able to really predict what's going to happen in response to one of these interventions that we hypothesize should help corticospinal function. So one of the issues surrounding TMS application that might be leading to this really variable responses across people is that usually we deliver brain stimulation or transcranial magnetic stimulation to the brain in what we call an open loop manner. And so this just means that we basically don't really consider what the brain is doing when we're giving stimulation. We just sort of deliver stimulation when we feel like it, and then we see what the response is. That can be problematic though, right? Because we know our brain activity isn't always at the same exact state. Our brain's always sort of fluctuating, even at rest, between high excitability states and low excitability states. So the idea is that maybe this open loop stimulation, by giving TMS during all these different brain states inadvertently, we're actually sort of shooting ourselves in the foot so that we're not able to get the effects that we want to get. One way we can account for this is by using closed loop brain stimulation. So the idea here is that we use something about the brain state to inform when we give brain stimulation. And so this leads to the very interesting question is if we're trying to account for brain state so that we're only giving TMS during some specific brain states that we think are particularly useful, how do we choose that state? There's many things that our brain does, of course, right? It's a very complex organ. How do we decide when we should give TMS? And so that's going to be part of what I'm talking about today. So if we're thinking about an ideal brain state where we would deliver TMS, there are a couple characteristics we wanted to show. One, because TMS is a non-invasive measurement, ideally we would be able to use another non-invasive measurement to deliver brain stimulation. We wouldn't want to start recording invasively when we don't have to. We also would want this brain state to be very temporally well-defined. So this just basically means that we know with pretty good confidence whether or not that person that we're testing, their brain is in a specific state or not. So it's easy to tell. And also we wanted to reflect something about the functional state of the human motor system, especially the corticospinal tract, because that's the tract that we're really interested in promoting function within. So if we choose a brain state that reflects something about that corticospinal tract, we probably have a better chance of modifying the function of the tract. And so this leads me to some of the technologies that we might want to use when we're looking at closed loop brain stimulation. So non-invasive measurements can be nicely done with electroencephalography, which is basically just putting all these little electrodes on somebody's head and recording their brain waves. These brain waves, as shown here, are actually really nicely temporally well-defined. So they have really excellent temporal resolution, meaning we can record this brain activity on a millisecond by millisecond basis, which gives us a really good way of knowing whether or not the brain is in a given state at any given time. And this leaves us with this third criteria that we want to address, whether or not we can reflect the functional state of the motor system within these EEG or electroencephalography signals. So we know from a whole bunch of previous work that neural oscillations reflect rhythmic electrical activity that's generated by the central nervous system. And here's just a figure from a review paper showing kind of what that activity might look like in a raw trace of EEG. And these neural oscillations can be described, among many other properties, by three main ones that I'm going to talk about today. So first we have frequency, then we have power, and then we have phase. So frequency just means how frequent or how rapid those oscillations are occurring. So both of these oscillations here are 10 hertz frequency oscillations. They're both going up and down at 10 hertz. However, they do differ in one main way. One is that the oscillation on the top is what we call a high power or a high amplitude oscillation, meaning that the size of these waves is pretty big. And then also on the bottom, we have the opposite. So the size of the waves here is clearly much smaller. This is something we would call a low power oscillation. And then also we have phase. So we can look at different parts of this oscillation and break it up into different phases. So at the very top or the crest of the oscillation, we have what I tend to call a peak phase of the oscillation, which makes sense. It's kind of the top. And then as well, we have at the bottom this trough phase, or sort of the negative going part of the oscillation. And what's important here is that you can actually measure phase in both high power and low power oscillations. And so you can see, even though these oscillations are different in their power, they do still have peak and trough phases. And so how do these relate to our motor system? So when we use EEG to record these neural oscillations, we typically see in sensory motor regions that they're oscillating between 8 and 30 hertz. And we can functionally subdivide these oscillations into the 8 to 12 hertz range, as well as the 13 to 30 hertz range. And these both functionally relate to movement in terms of how they modulate their power with movement. So how big those oscillations are changes with movement. So normally at rest, we have a healthy person just sort of relaxing, hanging out, and you can see that their oscillation is nice and big. So we would call this a high power oscillation. And then as soon as they start to prepare or plan a movement, the oscillation will decrease in size. So it will become now what we call a low power oscillation. So you can still see it, it's just not as strong. It will remain in sort of this low power state during movement execution. And then as soon as movement is done, it'll sort of bounce back up to its resting high power state. So this modulation of the size of these oscillations tells us that these mu and beta oscillations are nicely related to voluntary movement. And so they might be an interesting aspect of the brain to target with this closed loop simulation I've been talking about. So I'm going to talk, now that I've gone over a little bit of the background, I'm going to talk about three main studies today. So the first is looking to establish the contributions of oscillation phase and power to cortical spinal function. So the function of that cortical spinal tract I showed you guys earlier. We also are going to look at some work that I did where we looked at contributions of brain activity during different neural oscillation phases to skill learning. And then we're also going to examine, and this is some brand new data that I actually haven't presented before, so I'm looking forward to sharing this with you guys. But this is a study where we looked at the role of neural oscillation phase in simple movements. All right, so going into the first of those studies where we look at power and phase and cortical spinal function. So here I have a figure from Saskia Hagens. She did this study in non-human primates, and what she did is she recorded single neuron spiking rates intracortally in the motor cortex of non-human primates while they were performing a tactile discrimination task. And then in addition to recording those single neuron spiking rates, she also looked at the oscillation phase in those same regions. And then she sorted the spiking rates based on the phase of the oscillation of the local field potential. And so what she found was this really nice phasic modulation of firing rate. And you can see here at the peak phase, this is an alpha oscillation or a mu oscillation between 8 and 12 hertz. At the peak phase, the spiking rates were much lower than at any other phase, whereas at the trough phase, the spiking rates were much higher. So we see that these peak phases might reflect a low excitability, low spiking brain state, as well as these trough phases reflecting a high excitability, high spiking brain state. But what was particularly interesting about this study is that this pattern was really only present when the monkeys were relaxing. And then as soon as they started to begin performing a task that's known to weaken these oscillations, we saw that this pattern of phase-dependent spiking actually kind of disappeared. And because we know that movement and engaging in this task desynchronizes or weakens these oscillations, this led me to the question of if sensory motor neural oscillation phase and power interact to shape corticospinal function. And so this is the thing that I wanted to address in the human using a combination of transcranial magnetic stimulation and EEG. And so we designed a study to do exactly that. So we brought people into the lab. They came in, we set them up with EEG and the EMG recordings. And then afterwards, we had them just rest quietly while we recorded 5 minutes of resting EEG. And so their eyes were open, they were just relaxed, but we weren't giving them any TMS during this time. Afterwards, when they finished this resting recording, we completed the TMS portion of the study. So this was 600 single TMS pulses being delivered over about an hour or so. And then in response to each of these TMS pulses, we measured the size of the motor evoked potential, which tells us about the function of the corticospinal tract. And so I want to note here that this is actually an open loop TMS study. I know I spent the beginning talking about closed loop TMS and its benefits, but before we can really go into a closed loop study, we need to understand how the brain states contribute to corticospinal function. So what we did is after we recorded all of this data, we went back and we sorted trials into those in which TMS occurred during a peak phase. So here, around 90 degrees of the oscillation. And then trough phases, which were clustered around 270 degrees of the oscillation. And so we grouped all the trials occurring during each of these phases, and those are the ones that we looked at for the study. So because this is open loop TMS, the first thing we had to do was determine if our procedure for sorting those trials into peak phases and trough phases was actually working. The reason this is kind of critical is that TMS, when you give it, especially over top of an EEG cap, you're going to get these really big spiking artifacts in the EEG signals, and you sort of need to be able to handle those and remove them without distorting that phase information. So the figure I'm going to show you next is basically validating that when we looked at those peak and trough trials, we were looking at the correct ones. So what we did is we came up with a sorting procedure that allowed us to sort trials into those in which TMS occurred during peaks and TMS occurred during troughs. And I'm not going to go into the signal processing issues around that. But then we looked at the actual phase at which those pulses came. And we were able to see that those pulses actually did occur around 90 degrees for the peak trials, and they did occur around 270 degrees for the trough trials. So this tells us that we were accurately sorting our data into peak and trough phases. We also looked at the EEG activity at the time of TMS delivery for the peak trials and the trough trials. And so here we delivered TMS right around here for each subject because we were recording from the left hand. And you can see that the peak phases were associated with localized positivity over that same area that we were stimulating, as well as the trough phases being associated with localized negativity over those same areas. Finally, we also looked at all of the trials leading up to the TMS pulse. So that's what this black line here indicates. We averaged them all for the peak trials, and we see that when we do that, that TMS pulse is coinciding with that peak phase, which tells us it's working properly. As well as when we sorted all the trough trials and we averaged those, we saw that TMS was again occurring during the trough trials. So all of these data show us that our sorting procedure worked properly. And this is important. This is a quality step to make sure that any conclusions that we're making based on these data are correct because of how we're actually analyzing it. So now that we've gone ahead and confirmed that this procedure is working properly, we can actually look at the results. So here we have the relationship between pre-stimulus mu power. So this is the size of the mu oscillation leading up to each TMS pulse, so pre-stimulus. And then we have the MEP amplitude, so the size of the muscle twitch evoked in the periphery by TMS. And in the red, we have all of the subjects' data for those trials in which TMS occurred at peaks. And then in the blue, we have the same except for those trials in which TMS occurred at troughs. And when we look just at these thin lines represent individual subjects. You can see that the individual subjects show sort of variable relationships between power and the MEP amplitude for the peaks. And you can see some of them are flat, some of them are going up, some of them are going down, but all in all, at the group level, this sort of averaged out to a flat relationship. So no clear relationship between mu power and MEP amplitude when TMS was delivered during peak phases. However, we see the opposite when we look at the trough phases. So here you can see almost every subject is showing a positive relationship. There's just maybe one or two that are going the opposite way. And when we look at the group level, that averaged out again to a positive relationship between mu power and MEP amplitude, but only for the trough phases. And so when we look at the group level data without all these little individual lines, we actually saw this nice phase by power interaction. So if you look again, here we see this peak phase, we see no real relationship between mu power and MEP amplitude, but then we see this positive relationship for the trough phase. And this again was manifested as a significant phase by power interaction. So what does this mean? We talked about whether or not these two interact. That was something we were interested in pursuing in the study, but how do we interpret this, right? So first of all, we can infer from these data that the effect of mu power on cortical spinal function occurs through mu troughs. So mu peaks aren't really contributing to that relationship. For this reason, we think that maybe mu peaks are reflecting a neutral excitability state that is not affected by the mu power. And that in combination with some previous work in the visual perceptual domain that has shown a similar sort of phase and power interaction, these results suggest that maybe these phase power interactions are a general mechanism that coordinate brain function across different neural systems like vision and movement. And the really interesting thing for me that came out of this is that basically we showed that mu peak and mu trough phases reflect different physiological states. And given these differences in physiology, to me, this suggested that maybe there are some differences in how they contribute to motor behavior, which is what I'm going to talk about in the next study. So this was the main question of the next study, as I said. Do mu peak and mu trough phases differently contribute to skill learning? We're interested in skill learning because after strokes, people often have to recover many of the skills that they've performed in daily life, sort of using a new body and new body dynamics. And so skill learning is sort of a way of evaluating how this process works in the intact brain so that we can better understand it in the post-stroke brain. And so here we finally were able to apply this closed loop TMS, closed loop simulation that we've been talking about so far. So we designed a study where, let's start with panel B here, it's probably easiest. So this was a 2-day study. Healthy people came to the lab on day 1 and they completed a short resting EEG recording where we calibrated the closed loop TMS system. We also then trained them to perform a motor sequence learning task, which I'll talk about in a second. And then once they finished practicing that task, we delivered phase-dependent closed loop TMS. So we gave TMS during peak phases, so you can see it sort of coincides with the peak here, trough phases, or we gave sham placebo TMS during random phases. And the important thing to note about this control group is that basically this group should not show any changes in motor skill learning. And so this is sort of a reference group of how normal learning proceeds in the absence of any active intervention. So after we gave all of the phase-dependent TMS, we brought the participants back the next day and they performed that sequence task again for us so that we could evaluate how well they learned the task. And the crucial aspect of the design here is that we think skill learning stabilizes or consolidates during this window of time after initial practice. And so by delivering TMS during this time, we can sort of interfere or modify that process. And then if peak and trough phases are differently contributing to skill learning, then we should see different behavioral performance on day 2 between those two groups. And so all this said, let's talk a little bit about the task. So we had, again, healthy participants perform this task. This was a explicit motor sequence learning task. Those healthy individuals saw a ready screen for 3 seconds, followed by a go cue, and then they saw a sequence of numbers presented on a computer screen in front of them. Their job was basically to reproduce this sequence as quickly and as accurately as they could using their non-dominant left hand. And then as soon as they did that for 10 seconds, they rested for 10 seconds, and then they went all the way back to the beginning and repeated it. And so this whole thing is one trial. And during this sequence task, the participants performed 36 trials. All right. And then, one more thing I want to say is that we did deliver this TMS to the contralateral hemisphere over the motor cortex to the hand that was performing the task. So we think we were targeting this representation of the movement task itself. All right. So because we're doing closed loop TMS now, again, we need to check that our targeting of the different phases during closed loop TMS is working properly. So this is a pretty technically demanding procedure. And we basically analyze EEG data as it's coming in, and then we use that to tell the TMS device when to give stimulation. So that can be complicated, and we need to make sure it's working properly in our participants. So on top here, I have resting EEG data from all 3 groups, participants in all 3 groups. And this black line indicates a time point where we had the closed loop analysis algorithm identify the phase that we wanted to deliver stimulation during, but it didn't actually deliver stimulation. So just sort of send a marker to the EEG system. And then we looked at that after the fact to make sure that where it was marking in the data, that was where we wanted it to be. So on the top, we have all of the data for the peak group in red, averaged across all attempts, all of the trough data in blue, averaged across all attempts. And then we have the green data, which is the sham control group. And then we have the exact same pattern of data in the bottom figure. So this is the raw figure the raw data that has not been filtered at all. And this is the data that has been filtered. And in both cases, you can see that when we're trying to target the peak phase, the trough phase, and random phases, we're doing so pretty well. The reason this green line for the random phase is flat is because when you target random phases, all of that oscillations, they just sort of average out to a flat signal. So this gives us pretty good confidence that we're targeting it the way we want to. We also looked at those same little histogram plots that I showed you earlier, and we see the trials in which we were trying to target peak phases, we did so pretty accurately. Same for trough phases, we did so pretty accurately. And then when we tried to target random phases, we see that it's pretty evenly distributed around the circle, suggesting that we weren't specifically biased to one phase or another. And then finally, we looked at those EEG maps during targeting of peak phases. And you can see we have this localized positivity over the targeted area. For trough phases, we have this localized negativity over the targeted area. And then for the controlled or the random targeting, we have no real positivity or negativity over the targeted area. And each of these little circles just reflects the channels from the EEG cap that we use to extract this rhythm. So that's all that is there. And then also we looked at the power, so the amplitude of the signal leading up to this targeting in each group. And we show that, so this is a violin plot. So these X's reflect the mean, and then each of these individual dots reflect an individual subject. And we see that regardless of whether we were trying to target peak phases, trough phases, or control phases, that power leading up to that in the mu range was similar across groups. Okay, so now I've shown you that our closed loop stimulation approach was working properly. So let's get to the interesting stuff, the behavioral results. So here are our learning curves. We see day 1 here and day 2 here. And on day 1, the x-axis, we have the number of trials, and then on the y-axis, we have the number of correct sequences. So this is our dependent variable or our outcome measure for this task. And we can see that all three groups on day one, they're acquiring the skill pretty similarly. They're all improving. Whoops, where'd it go? They're all improving with practice to a similar extent. And then after they completed performing the task, we gave them this phase-dependent TMS intervention. And this is just a little inset showing the targeting of peak phases, trough phases, and random phases to sort of remind us that this is working well. Then the participants went home, they went about their daily lives, and they came back the next day for us. And when they came back the next day, we started to see the groups diverge a little bit. It's not super easy to see here, and we'll unpack this in a second, but we can see that subjects in the blue, which is the trough group, tended to perform the best, followed by those subjects in the control and the peak group. So what does this look like in terms of actual skill learning? So if we look at the comparison between the very beginning of day one and the end of day two, we see that subjects in all three groups improved the number of correct sequences that they could type to a similar extent. And then this is reflected in the violin plots again. So this just shows us that there's no changes or differences between groups and how well they learn the task. This helps us know that we can compare day two performance fairly across groups. Then we looked at offline changes. So this is the difference between the end of day one performance and the beginning of day two, or all of day two performance, excuse me. And we look at the end of day one and the end of day two. And if you look at the trough group, you see that it has this nice steepish slope. But when we compare that to the control group and the peak group, those have much shallower slopes. And this actually was present as a group by day interaction. So this means that the participants in the trough group improved more from the end of day one to day two, which surprisingly was, or nicely, was when we were delivering this intervention, than the other two groups. And this was present when we look at the data this way as well. We can see that the participants who received stimulation during troughs showed better learning or offline learning performance than those who received sham control stimulation. The subjects who received trough stimulation also showed better offline learning than those who received peak active stimulation. And then the peak group and the control group actually learned offline exactly the same. So this tells us that delivering TMS during these trough phases, which we talked about in the previous study, seems to facilitate this offline learning process. So what do we make of this, right? First of all, we know from these results that delivering closed loop TMS to human motor cortex during mu trough, but not mu peak phases, improved this offline learning process. And what does this mean? This is the first time that we've shown the use of closed loop TMS to improve a human motor behavior, specifically skill memory, which I think is pretty interesting. But at a mechanistic level, we have sort of two possibilities that we need to consider here. The first possibility is that delivering TMS during these trough phases increased motor cortical plasticity, which then the plasticity itself drove the better offline learning that we saw. Or the alternative is that because we're giving TMS in this phase-dependent manner, it's possible that the learning mechanism or the representation of the motor memory is only really active in the brain during these trough phases. And so we can only interact with that motor skill representation during those same phases. Crucially, it's very hard for us to distinguish between these two key interpretations. We do have some evidence that this is not the case, but we are not able to right now definitively show that this is the case, and this might be some plans for future work going forward. But regardless of which mechanism is responsible for these behavioral improvements, we can conclude that sensory motor mu trough phases reflect these sort of brief windows during which we can non-invasively strengthen skill memories. And this provides us with some motivation for using closed loop TMS to improve motor behavior. All right, so I've talked about learning. I've talked about just sort of the function of that corticospinal tract. What about simple movements that don't require learning? So this is the more recent work that I've done over the last few months, and I'm going to present this. It's still very new, but I think it's interesting to think about. So we know from a whole history of motor neuroscience work that movements tend to be initiated when the primary motor cortex releases a motor command that tells the muscles to contract. So the motor cortex says, okay, spinal cord, tell the muscles that it's time to contract so we can perform whatever action we want. But we've been talking about how these oscillations seem to coordinate motor cortex activity in terms of how they control their spiking, cortical spinal activity, etc. So this raises the interesting question is, are motor commands released during certain phases of sensory motor oscillations? And so to address this, we designed a very, very simple study actually, which I'll describe here. Basically, the rationale behind this is that if voluntary movements or the motor commands that produce voluntary movements are preferentially released during voluntary or during specific oscillatory phases, then if we let people just sort of choose when they perform a simple movement, those movements should sort of cluster near those phases. And so we designed a really kind of boring, straightforward task where we had people view a series of pictures on a computer screen. Basically, they saw a picture and then they got to look at that picture for as long as they wanted. And when they were ready, they just hit a button on a computer keyboard that said, okay, we're going to go to the next picture. And so they just did this for a while. And then before that, we actually measured their corticospinal or corticomuscular latency. So this is the latency from when we give a TMS pulse to the motor cortex to when we start to see a motor evoked potential originate in the target muscle. And the rationale here is that this latency reflects the time that it would take for a motor command to travel from the motor cortex to the periphery of the muscle that we're interested in. And so we looked at the EMG activity while subjects were performing this very, very simple key press task. And we basically identified the time where EMG activity started to be evident. And then we stepped back by the MEP latency itself. So this is the time that movement began. And then we estimated that this motor command release that led to that movement occurred before that by the MEP latency. So we basically identified the MEP latency, identified the EMG onset, and then we just subtracted the EMG onset, or from the EMG onset, the MEP latency. And so from this, we can get an estimate of the oscillatory phase during the time of motor command release from the motor cortex. Just checking on time, making sure doing okay here. All right. So here is some data from a representative subject during this study. So the black traces reflect the EMG activity, and the blue, reflect the EMG onsets that we visually identified. And this here reflects mu activity between 8 to 12 hertz, and this here reflects beta activity between 13 to 30 hertz. And then the red lines reflect just stepping back by the MEP latency to the time that we think the motor command would have been released to produce the self-paced movement that was associated with this EMG activity. So what we did is we looked at this in each person. We identified the individual frequency of oscillations for each movement, and then we calculated the phase at which the motor commands we thought were released during those movements. We averaged them across all subjects, and then we plotted them along in the phase space and along the oscillation. So here, this is the optimal motor command release times across the oscillation phase for the mu range. And we see that each of these dots reflects a different subject. And so if there is some systematic relationship between oscillation phase and motor command release, then subjects should kind of tend to cluster on a specific part of this oscillation. This isn't what we see here. So we actually see that those motor commands were released pretty evenly, depending on the subject, across the whole oscillation phase. And this is just the same data presented in a different way. I know it does look like there's more motor commands being released near the trough phase here, but that's actually not the case when we do that statistically, and this was not significant. However, when we look at the beta oscillation, we see something pretty different. We actually see that motor commands tend to be released around the peak of this oscillation. So this is different from the previous work in a couple ways. One is that it's the beta oscillation that's showing us the interesting results, as well as it's clustered near the peak instead of the trough. And so this was significant. Subjects tended to produce their movements or release the motor commands that led to their movements during this peak phase at an average phase of about 120 degrees. And what's cool about this is if we go back to the literature, there's some early work from the 90s that suggests that these EMG bursts are locked to beta trough phases, which is sort of at first might contradict this finding here where we see the motor commands being released near peak phases. But if we think about it, it takes time for the motor commands to come from the motor cortex to the spinal cord and the muscle. And so if we do the math on that and we sort of back calculate, in order for EMG bursts to be locked during trough phases, we would have to release the motor commands producing those EMG bursts around peak phases. So again, how do we interpret these results? So first of all, the estimated time of motor command release from the primary motor cortex occurred just after the peak phase of beta oscillations, around 120 degrees. And what's cool about this is that it sort of matches up with the timing of EMG bursts in the beta phase, but it also coincides with the beta phase that shows decreased M1 spiking. So beta peak phases are also associated with reduced motor cortical spiking. And so what do we think this might mean? This might mean that the decreased background spiking that occurs at beta peak phases could allow motor commands to be more efficiently transmitted from the motor cortex to the spinal cord by improving the signal to noise ratio. So you can sort of think of the background spiking as the noise and the signal as the motor command. So you can increase the efficiency of motor command release by either increasing the size of the signal or decreasing the size of the noise. And so if we couple those motor commands to a time when noise is decreased, then we have an improved signal to noise ratio. And so the conclusion here is that motor commands preceding a self-paced, simple movement are released during optimal phases of beta activity. And this is the first demonstration of that process of motor command release, possibly following a rhythmic pattern. And so all of these results, I think, inform us on some of the basic mechanisms of motor control. First, we showed that the mechanisms of corticospinal function vary depending on the neural oscillation phase. And this is particularly present within the mu range. I did also have some data that I didn't show that showed that this was not the case for beta. In addition, we showed that TMS improves motor learning only when it coincides with these optimal mu trough phases, and that motor commands preceding simple voluntary movements tend to be released at these optimal beta phases around about 120 degrees. So all in all, we showed this in the corticospinal range, in the skill learning range, and then as well in the simple movement range. And this suggests to us that the neural mechanisms underlying control of human movement are actually cyclic, that they vary from phase to phase, that these mechanisms that support a whole host of things that we need for voluntary movement are not actually continuous over time, but they sort of occur in a pulsatile fashion during these optimal phases of oscillations. And this leads into sort of the long-term goal of my work, which I'm still building towards, is that delivering brain stimulation interventions during these optimal phases could theoretically improve its therapeutic effects. So the idea is to use this information that we've learned about how movement is controlled in a cyclic manner to design neural stimulation interventions that respect that cyclic organization so that we can have stronger effects. And this leads me into sort of the long-term goals of my research program. A lot of what I talked about today falls under the first part of it, which is to seek understanding of how neural oscillations support normal and abnormal movement, and then to develop closed-loop brain stimulation interventions that target the parts of the neural oscillations that we think are important to movement. And so this was addressed a bit in the context of the second experiment I showed you. And then finally, long-term is to use these interventions to help people who have had a stroke or other types of neurological damage regain some of their motor function. And I think we're on a pretty good time to end here. I just want to thank several of the people who contributed to this work. Like I said, I completed a lot of this work in my postdoc at NINDS in Bethesda, Maryland. A whole host of people there helped me with that. Very supportive people, which was great. And then also we had collaborators in the University of Tübingen in Germany, Ulf Siemann and Christoph Renner, And then I also want to thank my funding source, which was through the Intramural Competitive Fellowship Program at the NINDS. And I'm happy to leave this up. I'm going to stop sharing though, I think.